Welcome back, everyone. Professor Hugh Pennington is 82, a microbiologist of distinction, an expert on virology, food safety and infectious diseases. And he is Emeritus Professor of Bacteriology at the University of Aberdeen. Outside academia, he's best known as the chair of the Pennington Group inquiry into the Scottish E. coli outbreak of 1996, and as chairman of the public inquiry into the 2005 outbreak of E. coli 0157 in South Wales. He's a regular contributor to news and current affairs programmes on television and radio, who seek out his expertise on matters of public health. Please welcome Professor Hugh Pennington.
Hello, thank you very much for that introduction, and thank you very much for asking me to speak to Rotary. I'll start off by talking about June Almeida, who I knew in the 1960s. I'm that old, and she was one of my most important mentors when I was training as a virologist, doing my PhD on virulence in viruses. And June was an electron microscopist. She was born in Glasgow. Her father was a bus driver. She left school at 16 because there was no money for her to continue her education, although she won a prize for science at school. And she went to work as a lab technician at the Royal Infirmary in Glasgow, moved to London, married an artist who came from Venezuela, and that's why she's called Almeida. And they emigrated to Canada, where she started work at the Ontario Cancer Institute, working as an electron microscopist. And June had very, very green fingers and became an author on papers when she was still a technician in the labs in Canada. They were less fussy in Canada about your standing and status. They were more concerned about what you did rather than who you were or where you'd been and all that kind of thing. And she was headhunted by my new boss in London at St. Thomas's Hospital to be an electron microscopist there. And that is where she discovered human coronaviruses. She'd been sent some samples from the Common Cold Research Unit on Salisbury Plain, run by David Tyrrell. And they'd had great difficulty trying to work out what these viruses were. They were common causes of the common cold, but they were very difficult to grow in the lab. And June put them in her electron microscope using a technique that she pretty well invented herself, which made things much simpler. And bingo, there were these viruses with, well, you know, this is what they look like, isn't it? And this is not a model of a coronavirus. This is actually a model that was used to teach medical students in Aberdeen in about 1910 about antibodies. And these are the antibodies coming out of an antibody producing cell. But it uncannily looks like a coronavirus of the sort that June discovered. Now, they remained curiosities for years. You know, who's interested in the common cold? There was no treatment for it. So there was absolutely no need to have a diagnostic test. And apologies for my phone going. That's it. Somebody else has answered it. But until basically later, they became important as human viruses, although June had already seen them in diseases caused by coronavirus in animals. There was infectious bronchitis in birds, and there was gastroenteritis in pigs. It's quite a bad disease for piglets. And she'd seen these before, and she tried to publish pictures of them, coronaviruses in animals, and the paper had been rejected because they just saw these are very bad pictures of influenza. And June didn't hold any grudge against the referees. I mean, you just regard them as rubbish, but there it is. The picture changed completely in 2002. Actually, it was 2003, but 2002, SARS appeared in China. And the Chinese screwed it up in a magnificent way. The virus had appeared in November, or at least cases of pneumonia caused by the virus had appeared in November, and they kept shum about it. And they didn't tell World Health until February the following year. And then World Health set up a team of virologists, worldwide international team, and it was run by a guy who I knew who said, one of the things this team must do is not jump around claiming priority. You know, we were the first to discover this virus and all that kind of stuff. And they had an agreement that they would work as a collaborative team. And very soon afterwards, it transpired, yes, this was a coronavirus, as basically using June Almeida's method in electron microscope, a virus with knobs on it, with, you know, knobs on the end. And it was new because they looked at the sequence of the genome, and it wasn't anywhere closely related to any of the other coronaviruses that were there. And the SARS experience was, of course, we escaped it in the UK. We had one or two cases only. But one very peculiar thing about SARS, at least it seemed so at the time, was that you got these super spreading events. One man in a hotel in Hong Kong infected about 70 people, and they hadn't really been very close to this guy. They'd walked past the, you know, they'd been in the same lobby of the hotel and all this kind of thing. And many of these cases, before they fell ill, had gone back to Vietnam and Canada and Taiwan and Germany. And so the viruses that were actually looked at by this team were from those countries. And they were all the same, and they were the SARS virus. And World Health cranked into action in April and May that year. And by July, they could say it's been sorted. The number of cases had disappeared. There was just the odd one case after that. The virus had basically become extinct, except in one or two labs. And of course, that's been our hope with COVID. Hasn't happened yet. Whether it will or not, I will perhaps talk about it in a few more minutes. But SARS it had a very big wake-up call for the authorities in the US and for the authorities in Taiwan and the authorities in Vietnam. There's this saying, of course, we must learn lessons. Well, I'm cynical enough to say, yes, we learn lessons and then we forget them again. You know, we have a propensity to forget them. You know, I was introduced as chairing an inquiry in 1996 on an E. coli outbreak in Scotland, and then a public inquiry in 2005 for another E. coli outbreak. And what I concluded in the public inquiry was that lessons had been learnt and then forgotten, because the same things went wrong the second time in Wales as it happened in Scotland in the late 1990s. So that's something we must remember, that we can have as many inquiries as we like. I'll talk about those in a minute, but we have to have a much better memory about these bad things that have happened. Now, countries like Taiwan and places like Singapore and Hong Kong had learnt the lesson from SARS, and so had the Chinese. It was a grave embarrassment to them. And, you know, the fact that they kept quiet about it, eventually they had to come clean because the virus had been found and the virus had spread to other countries. And the political result of that was that they sacked the mayor of Beijing and also the health minister. And you know, I don't know whether anything nasty happened to them or not, but anyway, they were fired. You know, that was made public. So some countries remembered SARS and had a system in place for when COVID appeared, bingo, you know, we could do something about it very quickly. And the Chinese did. The first cases of COVID appeared in January 2009, in December 2019, a handful of unusual pneumonia cases. And although it hasn't been publicly admitted, I think they thought, oh, bloody hell, here's SARS coming back. And they did all the tests of the day. In fact, they did more than all the tests of the day. They did some pretty unpleasant tests on these patients, you know, sticking tubes down the back of their throats and taking material out of the lungs and so on. And basically with the idea that, yeah, this might be SARS again. So they did the same tests. They looked down the electron microscope, bingo, there was a coronavirus. They did gene sequencing, And no, it wasn't SARS at all. It was quite a different virus, one related to bats, but it wasn't the same as the bat viruses they knew about. So basically, they did gene sequencing on this this handful of cases, viruses from a handful of cases, and they had those done by the first week of January, and they published them internationally on the 12th of January, and then there was a test available on the 13th of January, because you can use the results of the gene sequencing to construct a test, the sort of test, the PCR test that everybody's familiar with, the test that's the gold standard test for looking for COVID. So the speed of light in terms of the response compared the Chinese response in terms of the response to SARS. Where the virus came from, of course, there is vigorous debate. And there are those who say, well, it was a virus that evolved naturally. And then there are those who say, oh, it was a lab escape. Well, it can't have been a lab escape unless it was already there to escape, to actually jump the hurdles to get out of the lab. So had the Chinese constructed it, in some devilish experiment and so on, I don't think we'll ever know. We'll never know. And a little bit of history is helpful here. I was indirectly involved in the last lethal case of smallpox in the world, which happened in Birmingham. And in 1978, there was an escape from a lab in Birmingham that was working on smallpox. And unfortunately, it infected someone in another part of the university who died of smallpox. And Janet Parker was the last human case of smallpox in the world to have a fatal outcome of her infection. Now, I'd been to that lab about 6 months before because I'd examined the PhD student who was doing the work, which was allegedly the cause of the outbreak. And when I got home to Scotland, I immunized myself, I vaccinated myself, because the lab was about to be closed by World Health because it wasn't up to scratch in terms of all the safety procedures and so on. Well, that was a lesson in that. I don't think the Wuhan lab has ever had that kind of constraint put on it. But at the end of the day, there was a trial to determine was the University of Birmingham liable for this frightful incident, because, well, Janet Parker wasn't the only person who died. The head of department, Henry Bedson, who I knew well, who I had stayed with 6 months before, committed suicide because he just didn't want to be the last virologist to have this horrible thing hanging around his neck. Anyway, the trial let the Birmingham University off because they just couldn't accumulate enough evidence to say how the virus had got out of the lab and got upstairs and infected Janet Parker, who was a photographer working in another department of the university. They just couldn't establish it. Now, that was after very many investigations where people were there on the site investigating by taking witness statements and so on. I think we're unlikely to get that level of detail from Wuhan. And of course, that's not the first time that we've had a lab escape of virus in the UK, never mind anywhere else. The most recent one, I think, was 2003, when foot and mouth disease got out of a lab in Surrey. And I think probably part of the problem there was maintenance going on at the lab. And as we found out in the Second World War, when they looked at aircraft, that they concluded, doing operational research, that maintenance can be bad for your health. You twiddle with things that are working okay, but you twiddle with them to make sure they're working okay, and you leave them worse at the beginning after your maintenance than at the beginning. So I'm a bit skeptical about we'll ever find out about the lab escape from Wuhan as being the reason why this virus got out and infected large numbers of people in China and then spread worldwide. All we can say about the virus is that it's very good at getting about. One doesn't have to be a scientist to put that out. It spreads very easily. It spreads by the respiratory route, which is very difficult to stop. because the virus can blow on the wind. It lasts quite a while on the wind. It's much less dangerous on objects, and all the emphasis on hand washing and cleaning surfaces and all that is, well, I suppose you could say it's necessary, but it's a bit misplaced. It's not going to stop the virus spreading. It's when you get a large group of people in a room, particularly if they're heavy breathing in the room. And we know there have been big outbreaks in choir practices. There was a famous one in New Zealand in a pub on St. Patrick's Day when people were invited to come in and get themselves shamrocked. And they did, and there were 70 cases, and then some poor unfortunate man died as a consequence of that. This was all in February last year. No, this is not recent. This is basically set the rules for how we cope with trying to stop COVID. It's based on these unfortunate experiences. And religion plays quite a significant role as well. There have been very big outbreaks. There was one in South Korea, thousands of cases from a big religious gathering. There was one in France very early on in February 2000, another big religious gathering where people, they sing and clap and shout and get enthusiastic in a closed environment. And it's probably played a big role in India because they had lots of religious festivals before their second wave took off. So those are the kind of meeting people together and having a lot of shouting. Shouting is very, very important in terms of enhancing the spread of the virus. So this is an unusual virus in the way we're handling it, because we're handling it using the full technology of molecular biology. And this is the first pandemic that we've been able to do that, because gene sequencing used to be very expensive. And sequencing even the virus gene took ages. It would take weeks and months to do one virus and find out what its genetic makeup was. Well, things changed around about 2008 when new methods came in called next generation sequencing. And before that, we had the Human Genome Project, which took many, many years to sequence the human genome. Many, many people were involved, and it cost more than $2 billion. So it took more than a decade, cost $2 billion. Now, with next generation sequencing, you can sequence your own genome, or rather, you can get somebody to sequence it for you in a day, and it costs less than $1,000. So there's been a fantastic improvement in speed and a fantastic reduction in costs in how we can do that. And we're applying this to COVID. And thousands and thousands, hundreds of thousands of COVID strains have been sequenced. We set up a program in the UK last March, March 2020, to do this. And we've sequenced more genomes, I think, than any other country in the world, including the US, which is a bit of a puzzle, but there it is. And we can basically find out where the virus strains have come from, because this virus, it doesn't mutate as fast as flu, but it does mutate at a reasonable frequency. It doesn't mutate as fast as flu because mutations happen when there's a mistake made in the copying of the RNA. And with flu, well, the copies survive. Well, if the virus survives with the mutation, because most mutations are dead end, the virus doesn't grow at all after it's got a mutation, but some do. With COVID, it has a proofreading mechanism which basically chips away at any unusual copy alterations, any mistakes, and removes them. And so its mutation rate is less than flu. Never mind, it mutates pretty fast. And there are, I don't know how many variants have been basically identified, but it's many thousands of variants have been identified. And using these variants, it's possible to say, yes, the first wave of cases in the UK, most of them didn't come from China. That's where we thought they might come from. And we'd locked up people who'd come back to the UK from Wuhan. They came from basically like Italy and Spain. And a lot of them came back from people who'd been on skiing holidays and picked up the virus during the skiing holiday, and basically introduced the virus into the UK when then it spread in the community. And we know that the main place where the virus spreads, apart from these events I was talking about, these giant events where you get a giant sort of type of spread, is in the home. The home is the place where the virus usually gets about. And it's obvious that if somebody brings the virus into the home, it has all the opportunities there of, you know, people talking and, you know, all that kind of stuff, and close proximity for ages. Length of time that you're close to somebody is important in determining the risk level as to whether you get the virus or not. So the virus got going after the first wave, mostly by being introduced from Europe, where it had already, how it got into Italy, which is where it first made its impact on intensive care, I don't think anybody really knows, but presumably some people had come back from Wuhan and brought the virus to Italy when it spread in certain parts of Italy and Lombardy and so on, not everywhere. So that's, we can explain how the first wave happened. And even in the second wave, quite a lot of those cases, not all, but quite a lot of those cases were people who'd come back from holidays to countries where the virus was still busy. And that's one reason why everybody now, the authorities are very, very concerned about foreign travel, because that's the way the virus comes back. Nothing new about that. That's how we used to see smallpox in the olden days. And I like to go back to smallpox because I'm one of the few people remaining who's actually worked with it on the bench in the laboratory. But smallpox, we eradicated it many, many years, early in the 20th century, essentially. But we still had outbreaks, people coming back from the Indian subcontinent in particular would start off an outbreak. They had a certificate saying they'd been vaccinated, but what the vaccination certificates didn't say whether it had been successful or not. And one also suspects there might have been a little bit of jiggery pokery going along with these certificates as well. So I think that comes into the thinking about, you know, vaccine passports, all very well, but they have their limitations. Smallpox doesn't spread anywhere near as easily as COVID. So the outbreaks we managed to control, although there's some quite big outbreaks. The big problem with smallpox, of course, we didn't have a lab test for smallpox. We didn't need one because people got spots. The trouble was that people with spots were sometimes misdiagnosed as having chickenpox. And that's how outbreaks very often started. Here's somebody admitted to hospital with quite severe chickenpox, so they thought, actually it was smallpox. And the people who usually fell ill were the doctors who hadn't bothered to keep their vaccinations up, and the nurses who hadn't bothered to keep their vaccination status sort of active, as it were. So there are lessons from history, even with viruses, which seem to be really quite different from COVID. So, you know, where are we going from here with COVID? Well, you know, the number of cases as it goes up and down. We bring in lockdowns, the number of cases goes down. The one big thing, of course, that's going to be different in the future from what happened last year is we have vaccines. And that's another example of modern molecular biology. Just as the test is utterly dependent on knowing what the sequence of the virus genome is, and that depends on having these modern techniques. The vaccines are very unusual in that you synthesize the bit of the genome that makes the spike protein on the outside of the virus, you know, this thing here. And that's the bit of the virus that sticks onto the cell to infect you. And it's also the bit that antibodies that you make, either when you've recovered from an infection or whether you've had the vaccine, they stick onto that and they stop the virus sticking onto the cells. So basically, you know, we virologists call that neutralization. It's not a very sophisticated term, but that's what it means. So the virus is dead for all practical purposes. And so now we've got a very significant minority remaining unimmunized, but the majority of the population are immunized. The virus is not going to go anywhere in them, except that no vaccines are 100% successful. I mean, there will be some people who've had their double doses who will still get the virus. We know that's been recorded for quite a long time, but they tend not to get anywhere near the same degree of illness as people who haven't had the vaccine, or before people had a vaccine. The messenger RNA vaccines are the ones that I'm really talking about, these brand new vaccines, where you make a little bit of chunk of the genome that codes for the spike protein and either stick that directly into the patient or stick it into another virus. And the one that's used is basically Oxford vaccine uses the adenovirus, which is a dead virus essentially, but it's a sort of carrier for a little bit of genetic information. And both of these vaccines are very successful. There's this very peculiar side effect with the Oxford one, which was entirely unexpected and wasn't picked up in any of the trials because it's so rare, where they get these sort of blood clotting problems, and nobody's managed to explain why that vaccine should do that. It's a great scientific puzzle. There's no obvious explanation why it should do that, but nevertheless, it does it very, very infrequently. So it's not really a, I mean, I've had that particular vaccine. I didn't get it. Well, that doesn't prove anything, does it really? But all I know is that, you know, I've had my 2 shots. So, you know, my daughter from Glasgow came and she's in the house at the moment. So, you know, that couldn't have happened a month ago, really, because I didn't have my immune status to the level that I would have been happy with as a virologist. The great advantage of these messenger RNA vaccines is that they're synthesized in the lab, or the bit of the vaccine that's the sort of the bit that's important is synthesized in the lab based on the genetic information, really going back to the Wuhan type of virus. And if we need to change it to make a new booster vaccine, if the virus itself mutates to form a virus that's resistant to the immunity that we have from the vaccine, it's a very simple job to make a new messenger RNA vaccine based on the sequence information. And we're doing lots of sequencing, so we'd have that information very early. And the only issue there is not making the vaccine, but it's making enough, because clearly there is a problem in the world in making enough vaccine to go around. And, you know, there've been all these disputes with the EU and all this kind of stuff, just because we don't have the capacity to make as much vaccine as we would like. And setting up a lab to do it is not trivial, because it has to be a lab that delivers a safe vaccine. You know, there were tragedies way back 100 years ago when vaccines were made in very dodgy circumstances and caused lots of mischief. That doesn't happen nowadays, because if you want to look for somewhere that's as regulated as it could be, go to a vaccine lab. It's regulated not only by the external regulators, but by the people who make the stuff. It's not in their interest to put out a dodgy product and so on. So I'm optimistic that we'll be able to cope with any kind of variants. Now, I said earlier that there are thousands of variants have been identified, and their main utility is tracking where a virus has come from. Because if you find variant A in a group of people, and you know that this variant was first described in some other place, well, that's probably where they got it from. Not 100%, but pretty good. It's like fingerprinting, really. And of course, the other utility of looking for variants is that some of them might make the virus a bit hotter, as it were. There's a big problem with getting that sort of information. As we can see at the moment, there's some dubiety about how hot these viruses are, these variants are. You know, is it more transmissible by 20%, 60%, 100%, or whatever? The problem is that these, we can't predict from where a variant takes, you know, where a variant occurs in terms of where the mutation is. We can't predict confidently that this is going to lead to more transmissibility. What we have to look at is this virus causing, is this particular variant causing more cases in a way that basically can only be explained by getting around a bit better than the original virus. And that's very difficult to establish, because the original virus gets around very well, thank you very much, to start with. And finding out that the virus is getting about a lot better is also complicated by the fact that it may well be other circumstances that are helping that particular variant to get about. Like, for example, in India, where there's pretty good sort of circumstantial evidence that religious festivals, election meetings played a big role in getting their current wave going. You can't just blame the variant for that. It may have played a role in being more transmissible. The only way you could really tell about these variants being more transmissible is to do volunteer studies. Take a group of people who have no immunity and infect them with the virus, and then another group of people infect them with the Wuhan virus and see what happens. And wouldn't be too happy about being a volunteer in any of those experiments. Well, certainly not me at my age anyway. But still, that's probably not going to be done. And it's still indirect evidence. And that the genetics comes before working out what the virus does, which is the other way around from what we usually do. We usually find that a virus is spreading, and then we can work out why from looking at the genetics. And so it's quite complicated, and that's why there's so much sort of if and but. We must wait for the evidence kind of talk at the moment with working out what's going to happen. Are we going to get rid of the virus? Don't know. Wait and see. I mean, there's been an enormous amount of jumping up and down about herd immunity and Dominic Cummings and all that kind of political stuff, because there was no doubt that early in the first wave, UK government policy was basically a sort of herd immunity policy, based on the fact that this virus basically only causes mischief in people over the age of about 40 and 50. And the older you are, the more likely you are to go to ICU, and the more likely you are to die from it. And it's unlike any other bug. No other bug has that kind of relationship where basically young kids, don't turn a hair with the virus. There's a very rare syndrome in some very young kids, but most of them get better from it. But as you get older, particularly after the age of about 40, you start to see mortality, and even at 40, it's a very low mortality. But at the age of 80, it's 20%. At 40, it's sort of less than 1%, and that kind of thing. And there's no other bug that behaves like that. The one or two others have a bit of a similarity to it. The one I like to quote is typhus fever, which was the sort of concentration camp disease during the Second World War and caused big epidemics in Poland after the First World War, and it was jail fever in the UK, in the 17th and 18th century, and it was the big killer in the Irish famine in the late 1840s, and that had a relationship a bit like COVID, except that it went for the elderly, but it also did go for young people as well. Not to anywhere near the same degree as the elderly. We've never managed to explain why that was, and I don't think anybody's come up with a satisfactory explanation with COVID. There are other risk factors for getting basically a hard time with COVID, and a very important one is obesity. Can't explain why that is. And other conditions that affect your immune system. Diabetes is a factor, but that's often associated with obesity. And also, people who've had organ transplants and so on, because basically they're permanently immunosuppressed, at least to a degree. And that seems to help the virus no end. Particularly people who've had, blood cancer type conditions. And then, of course, there's an ethnic aspect to it again, which again, nobody's really satisfactorily explained. It may be that, many groups of people, because of occupational exposure, have been more likely to catch the virus, but it may be more than that. And of course, other diseases have an ethnic sort of distribution, particularly high blood pressure, for example, is when you look at lab test results, you have to take that into account when you're interpreting the results. So, there are these unanswered questions. And of course, what the virus does, and I'll finish on this note as a happy sort of note, most people have a very mild infection. Many young people have no symptoms at all. but they're dangerous in the sense that they can spread it to somebody who might get serious symptoms. And as you get older, you get pneumonia, and then the organs in some of these severely infected patients fail as well. So you can get kidney failure and so on. And this is obviously meat and drink, let's put it like that, crudely, to people in the intensive care unit who are very familiar with handling patients, with those kind of complications and so on. We don't fully understand the pathology of why it's doing what it's doing, but we know enough about it to know that, for example, you can alleviate some of the more severe cases with steroids, dexamethasone. Nothing new about that. We've been using that for years to do that in those kind of patients, but you had to learn by experience to find that that worked for COVID as well as it works for other kinds of pneumonia. So that's where we stand at the moment. Whether we'll get anti-COVID drugs, who knows? Might not. Basically, will the virus disappear? Don't know. When we get a higher level of immunity, which is this herd immunity kind of idea, it might be enough to protect the whole population. The virus might die out in the UK. Nobody's really quite certain about that. The problem then would be importation. And of course, to end on a happy note, New Zealand managed to eradicate the virus, except they've had the occasional importation, and the same with Australia. But unfortunately, in both New Zealand and more in Australia, the quarantine hotels have turned out to be a bit leaky, that people have been behaving badly, and the virus has been getting out and causing mischief, which has a very major impact on those countries in terms of their response, because they want to keep the virus levels at 0. We're a long way from that yet. So on that note, I'll finish and answer any questions.
Thank you, Professor. That was a fascinating talk. Does anyone have any questions? Yeah, Melvin. You need to unmute yourself, Melvin.
As someone who spent about 25 years trying to get some sort of consistency in the manufacture of pharmaceutical drugs, you say about the ease of changing over vaccines, but I've heard that batches of vaccines could be not up to scratch. What's your view on that?
Well, I suppose that's more than a theoretical possibility, and they have to be checked to make sure that they are actually what it says on the label. I've not heard of any particular, I think there have been 1 or 2 anecdotes about that vaccines haven't really been as good. I think one of the bigger problems has been with the Chinese vaccine, which was used very extensively, has been used extensively in Chile, but doesn't seem to have had the same effect as the AstraZeneca and the Pfizer vaccines in the UK. I suppose it depends on having good quality management of your in your vaccine manufacturers to make sure that the batches are all the same and have the same constituents and all that kind of stuff. And I suppose when it's a new vaccine, well, we haven't built up a lot of experience of making it over a long period of time. So keeping my fingers crossed.
Thanks. David Fraser, please.
Oh, sorry, I pressed the wrong button. Yeah, sorry about that. This is more of a comment and a question, but I would be interested in what Professor Pennington has to say about this. At the beginning of last week on GMTV, there was a British scientist whose name escapes me. He was being interviewed, and he explained how he'd been heavily involved in research into HIV and how the virus that caused HIV had been modified to make it safer to work with. The same scientist made the statement that he had visited a lab in Wuhan and been told by the Chinese virologist who was showing him round that they had modified the coronavirus originating from bats so that it would destroy every cell in the human body. Now, as this scientist said last week, why would the Chinese do that?
Well, that's a very good question. Particularly, then let it out of the lab, and firstly infecting Chinese citizens. I think it sort of talks a little bit about the kind of issue of biological warfare. Well, let's stick with that, because what you have to do with biological warfare, unless you want to destroy the human race, is you want to have a weapon that will keep your population safe, but will do damage to the population of your enemy. Well, there isn't the Chinese at this time, I don't think, way back, 2019, which is presumably when the virologists are speculating about what happened. There was no vaccine and it hadn't been developed. And as I said, the Chinese vaccine is perhaps not up to scratch, relatively speaking, and so on. So it'd be a bit of a puzzle if somebody did that. I think the more likely thing would be that they were fiddling about with bat coronavirus. We know that they were collecting bat coronaviruses. The trouble about making it more virulent is we don't know how to do that. And I don't think the Chinese still know how to do that, because it's not a question of saying, we'll take that particular gene and we'll modify it in this particular way, and that will guarantee it to be a nasty. Nobody knows how to do that. And it's almost certainly it's a composite of the action of all the 10 or so genes in the virus that leads it to become nastier. So I think, he's putting a lot of emphasis on something that if the Chinese knew how to make this virus nasty, they should tell us how to do it, because that would be very useful information in terms of knowing how the virus causes illness and so on. And I don't think we're anywhere close to that. I'll just finish by talking about flu, because we've been looking at gene sequencing of flu for years, trying to work out why some flus are nasty and some are not. And we haven't succeeded, because it's a composite of all the genes working together, rather than saying, oh, this is the nasty gene. And all we have to do is put a few mutations in there and away it goes. Not as simple as that. So I put a fair degree of skepticism about that story, I would have to say.
Okay, thanks for that.
Thanks, David. Alex at Cowdenbeath, please.
Hi, Hugh. That was a tremendous talk that you gave. But my question is around whether or not we're going to have to be living alongside COVID from here on in. I've heard some talk about the fact that we may have to be having like an annual vaccination program, a bit like the annual flu jabs, the seasonal flu. What's your opinion about that? Is that a distinct possibility?
Yes, I think it is. I'm not saying it's going to happen. We don't know. It really depends on case numbers and how we're coping with basically controlling our borders, and how other countries are doing. Countries that we have a lot of trade with, I mean, a lot of personal movement with, would still be places that we'd have to worry about. Of course, we can't control what they're doing with their vaccination program and all that. But nevertheless, I think if it came to having to have another jab every year for COVID, because there would be this is basically going to be protecting the elderly. We needn't worry about the cases in the young people. It's the elderly, because only the elderly fall ill and die from it. I don't think that would be a bad thing, because at the moment the flu vaccine we get has got more than one virus in it. We could stick COVID into that as well. Okay. So you wouldn't be doing anything other than having a flu COVID jab rather than a flu jab. And all we can say about the COVID jab is that the COVID jabs we have at the moment work better than the flu jab. And I don't think putting COVID in with the flu would really have any negative effect either on the COVID immunity or on the flu. So I wouldn't see that as anything more than a reasonable response to the virus hanging in there for a long time. The hope is that we manage to get rid of the damn thing, but that's not for this year probably, because it's still busy in India and it's still busy in Brazil and worldwide and all that kind of stuff. And until we've got to the same position that New Zealand is in the UK, and other countries have got to that position, I think having a COVID jab annually, it would be a small price to pay.
Thanks, Alex. John Milne, please.
A short question, Professor. Long COVID, the concept of long COVID, is that unusual in other viruses, or is it so early on that what's going on can't really be detected or properly researched?
Well, nobody quite knows what's the cause of long COVID, and it's almost certainly more than one condition. There are people who get these sort of myalgic encephalomyelitis, type of conditions after an infection. Whether it's the virus hanging in there, I rather doubt, because that can be tested for. I've not seen any evidence it's the virus. I think it's more the impact of the virus on people, leaving them with fatigue and this condition unknown to medical science called brain fog, and so on. And until we can work out how many of the people are basically have almost a psychological response to the virus infection, or how many people have got some definite damage that's been caused by the infection, which is persisting. The only certain thing is that it's going to be very difficult. And the only other certain thing is that people who've been in the intensive care unit and have had a long stay there do get basically there's a post-intensive care syndrome, which is definitely due to all the horrible things that have been done to them to save their lives, being intubated, things pushed down your windpipe, you've been anesthetized for weeks and so on. And it would be a great surprise if that didn't leave an impact on people, particularly as the people who've had that, were very sick to start with, and also were often had other conditions wrong with them, which made them more likely to get the virus in the first place and have a hard time. So I think the post-intensive care thing is not long COVID. That's something else that we've seen with other conditions that drive you into intensive care. But long COVID itself is a big puzzle. And I'm not particularly optimistic that we'll come up with a simple answer, because we've had other diseases like that. There was a thing called the Royal Free disease, when lots of nurses from the Royal Free suddenly came down with a kind of long COVID. This was long before COVID appeared. Never worked out what that was due to. And there was a neurological disease in the 1920s, which left people with a propensity to get Parkinson's disease and so on. But we never worked out what the cause of that was. So many unanswered questions. Good for researchers in that there'll be research grants available. Not so good for the people with long COVID, because I think many of them will be told to pull up your socks and get on with it and all that. Whereas some of them are obviously seriously sick. They do have something wrong with them. So it's a very tricky scientific issue, which is not going to be resolved anytime soon.
Thanks, John. Final question from Brian Johnson, please.
Professor Pennington, thank you very much for your talk. I have family that live in America and have not seen them for 2 years now. And it used to be, like many Rotarians, the sort of person that would travel extensively and think nothing of it. I'm certainly not in any inclination to go and swim myself on a beach in Portugal, but I'm very desperate to get to see my family, who I've not seen for 2 years. One option that we're actively looking at the moment is whether we should go to Iceland. They could go and visit Iceland, and we could more easily visit Iceland, it seems, than America. What is your feeling about when international travel will begin to ease for us? And do you think there'll be restrictions there in terms of vaccine passports and the like?
I'd be very surprised if there weren't vaccine passports for entries to countries. Well, I don't know about Iceland, but they've coped reasonably well with the virus in Iceland. I haven't looked at their case numbers, recently, but I think they were pretty low. And of course, being an island gives you an advantage, particularly if you know where people are coming from. and you can restrict the entry of people coming from hotspots and all that kind of thing. I think going to Iceland would be a very good idea if they let you in. You know, it'd be like go to Australia, they won't let you in, and certainly not New Zealand, they won't let you in unless you're a citizen and all this kind of stuff. And even then, you know, you'd have to go into quarantine, and well, you know, and there's probably a waiting list to get there. But on the other hand, as you know, as say the European case numbers come way down, which let's hope happens, and the US, of course, has got a very vigorous immunization program rolling. You know, with, I mean, one thing you can say for Donald Trump, you know, he got the warp speed program rolling for vaccination, and you know, whether people are taking it up or not is another matter. But you know, Americans have usually been pretty good at taking up vaccines. I mean, there's Andrew Wakefield is out there at the moment, bloody hell, you know. But never mind. So I think that would be reasonably optimistic. But when this will all sort of ease up is very difficult. You know, wouldn't want to predict that. Wouldn't want to predict that at all, because you've only got to get another surge of cases somewhere, you know, and the authorities just clamp down, like, you know, UK, like we have on Portugal, for reasons which are not, you know, not entirely clear. And if I could throw in one extra thing, which is just a slight hobby horse of mine, is that I think the variants are being used as a bit of a stick to beat us over the heads with, to get us to behave ourselves. You know, it's not to say that they're not important, and it's not to say that they may be more transmissible, but you can say, maybe, you know, the US authorities would say, oh, you've got the Indian variant circulating in the UK. No, we're not going to give you a visa, kind of thing, you know. And we've got to cope with that. Nothing I can do about that, but, you know, bear that in mind, that there may be circumstances like that appearing that might throw any kind of well-laid plans, you know, into the bin, basically, because we don't know what new variants are going to appear, except we can be certain that they will. Whether they're going to be vaccine escape variants, you know, which get round immunity, it seems unlikely. But if they did happen, that would change the picture very dramatically. So that's a bad note to end on, but, you know, slightly pessimistic, but, you know, watch this space.
Well, Professor, thank you so much for joining us today and giving us this incredibly informative presentation. I'd like now to ask everyone to join me in thanking the Professor for giving his time to our conference today and really giving us an incredible insight into COVID. Thank you very much, Professor.
Hi, John.